Analytical balance and volumetric glassware is one of the most important equipment to use to do an experiment. Uses of this equipment is to get an accurate reading, solution preparation, and for glassware calibration. Plus, it's reliable experimental to work at the pharmacy and pharmaceutical science. In scientific research and industrial application, precise measurements are essential for making informed decisions, optimizing process, and ensuring compliance with regulatory standards. While doing solution preparation, volumetric techniques are applied to create the standard and working with exact concentration. Last but not least, glassware calibration are super important to do before doing an experiment. Before use, measuring equipment must be calibrated and verified that conforms to do the laboratory requirements for its measurement uncertainty.
Now, I'll be explaining the theory behind two essential tools, the analytical balance and volumetric glassware. Let's start with the analytical balance. This is a high-precision instrument used to measure small masses, often to 4 decimal places. It is essential for the accurate reading of drug ingredients, especially during formulation. To ensure reliable results, analytical balance are enclosed to prevent errors from air or dust. In pharmaceutical labs, they are mainly used for drug formulation, preparing standard solution, and quality control tests. Now, let's move on to volumetric glassware. These are tools designed to measure exact volumes of liquids. Some common types include volumetric flask for preparing solution, pipettes for transferring fixed volumes, burettes for titrations, and graduated cylinder for rough volume measurements. Volumetric glassware used in titration and dilution process, purity and concentration tests, and ensuring correct drug dosage. Together, both the analytical balance and volumetric glassware play a vital role in assuring the accuracy, safety, and quality of pharmaceutical products.
Now, I will explain about methodology of analytical balance. In the picture, there is an analytical balance and its part. It consists power button to turn the balance on and off, then mute button to configure the measurement conversion system and change the measurement if necessary. It also has tare button is used to rebalance the system and bring it to neutral zero. It also has level adjustment feet that enable the balance to be brought to the reference position, and they are movable. On the balance, it has display panel where it displays various information like results, errors, function settings, and functions in progress. An analytical balance must have balance panel where it holds the sample material for the mass measurement, and it comes with anti-draft ring. Lastly, we have glass door to protect the samples from outside factors that can compromise the precision. There are various types of analytical balance. There are two-pan equal arm analytical balance, single-pan unequal analytical balance, microbalance, and electronic single-pan balance. We're moving to the procedure to use an analytical balance. Firstly, we have to select the proper location. We have to avoid direct sunlight, a stable and horizontal location that are dust-free, refrain from touching magnetic or magnetic field generating objects, and clear of air current produced by ventilators, air conditioner, and open windows. Next, Leveling the analytical balance. On the level adjustment feed, it has air bubble indicator where it shows if the analytical balance is leveled to account for any slight deviations or tilts. Number 3, calibrating the analytical balance. The analytical balance must be calibrated for a sample to be precisely weighed, and these are the circumstances called for calibration: modifications to the usage location, alteration in the environment, and before each use. Next is weighing. The analytical balance must be preheat for an hour before using it. Set to 0 in the no load condition by pressing the tare button and place the weight board at the center of the weighing pan. Avoid putting things in the balance as it can contaminate the balance. Lastly, cleaning an analytical balance. We may use just a piece of soap wet, mild detergent coated cloth. Avoid using harsh cleaning chemicals and cut off the electricity and unplug the power cord. Make sure there are no liquid or dust gets inside the housing of the analytical balance. Now, we move to the factor that can affect performance of analytical balance. First, temperature can affect noticeable fluctuation on the results. Number 2, vibrations can alter the precision of the balance. Number 3, chemical reactions should always remain inert. Number 4, air current will miss alignment in the sensitive support. Number 5, the calibration need to be certified. Number 6, magnet may attract other elements. And lastly, fingerprint may alter the results altogether. Methodology of volumetric glassware pipette. There are various types of pipette. The most common one is pipette, electronic pipette controller, multi-channel pipette, pasture pipette, pipette controller, and pipette filter bulb. In the picture, we can see the pipette and its part. First, plunger. Plunger is used to aspirate and dispense the desired volume of liquid. Number 2, tip ejector button is to eject the used pipette tips. Third, volume windows that displays the set volume and the place values shown depending on the pipette size. Fourth, tip cone is where the tip is attached. And lastly, pipette tip is a piece of equipment made of polypropylene that has been molded into it and comes in contact with the liquid. Remember, the pipette tips must always be sterile before use. How to use a pipette? First thing first, we take a tip out of the tip box, then adjust the volume to be transferred. Press the plunger to the first stop, then place the tip inside liquid component that being sampled. And once the tip submerged, slowly release the plunger to draw up the liquid. Once it's in, draw out the liquid into the tube or container we need and depress the plunger completely until it stops at the second stop. To remove the used tip, we press the ejector button beside the plunger. Using a pipette can be tricky, so these are 3 precautions. First, fill a pipette to the brim and drain it against the container's walls to make sure all the liquid is completely drained out. Second, carefully remove the disposal pipette tips. And lastly, all the mechanical pipette equipment must be utilized.
Now let's move on to the results. This demonstration clearly emphasized the vital role of both analytical balances and volumetric glassware in pharmaceutical laboratories. Through our simulation, we saw that analytical balances are able to achieve high precision with minimal error. This is especially important when weighing active drug ingredients, when even small inaccuracies can affect the final product quality and safety. Similarly, we found that volumetric glassware such as pipettes, burettes, and volumetric flasks were effective in measuring and transferring liquids with high accuracy. This ensures correct preparation of pharmaceutical solutions. To summarize our finding, for the first analysis, analytical balances provide precise measurement while volumetric glassware supported accurate solution preparations. And for the second analysis, the correct use of both tools guaranteed the accuracy, reliability, and safety of the pharmaceutical process. Third analysis, and lastly, we observed the proper calibration and carefully handling are necessary to reduce error and maintain quality control. Overall, this simulation highlights just how essential these pharmaceutical instruments are in ensuring consistency and compliance in the pharmaceutical work.
Conclusion, the use of analytical balance and volumetric glassware is essential in pharmaceutical laboratories to ensure accurate and reliable measurements. Analytical balance provides precise weighing and pharmaceutical substance, while volumetric glassware allows for the correct measurement and preparation of liquid solution. Proper handling and regular calibration of these instruments are necessary to minimize experimental errors and maintain high standards of quality and safety. This experiment has reinforced the importance of accuracy, consistency, and good laboratory practice in pharmaceutical applications.